In this module, I will describe non-coding RNAs called Y-RNAs and tell you what we know about their cellular functions. The vast majority of cellular RNAs do not code for proteins and are thus referred to as non-coding RNAs. In cells, these RNAs are normally bound by proteins to form RNA protein complexes, or RNPs. Some of the major classes of RNPs in mammalian cells are shown here. In the nucleus, small nuclear RNPs carry out pre-mRNA splicing, while the telomerase RNP repairs the ends of chromosomes. The cytoplasm contains ribosomes, which carry out protein synthesis. In addition to the many RNPs whose function is well understood, there are some RNAs whose functions are only now being elucidated. One of these classes of RNAs, called Y-RNAs, is the subject of this module. Y-RNAs are non-coding RNAs, 65 to 115 nucleotides in length, that are present in most animal cells and also some bacteria. They are transcribed by RNA polymerase III, the same polymerase that transcribes transfer RNAs. Most species have multiple Y-RNAs. For example, there are 4 different Y-RNAs in human cells, called Y1, Y3, Y4, and Y5. Y-RNAs are shaped like lollipops. All Y-RNAs fold into conserved structures containing 2 distinct domains. One domain consists of a long stem formed by base pairing the 5' and 3' ends. This domain contains the high-affinity binding site for a protein called Rho60. These sequences are boxed here. The other end of all studied Y-RNAs bind what we call effectors, which are proteins that act on RNA, such as ribonucleases. The Rho60 protein is the major protein partner of Y-RNAs, and Y-RNAs from humans, mice, and bacteria are all unstable when Rho60 is absent. This shows a northern blot from wild-type mouse cells and tissues compared with the same cells and tissues from a Rho60 knockout mouse. In all cells and tissues lacking Rho60, Y-RNA levels are drastically reduced. So this leads to the question, what is the Rho60 protein? Rho60 is present in most animal cells. Some sequence bacterial genomes and some archaea. My laboratory collaborated with Karin Reinish's laboratory at Yale to obtain several crystal structures of Rho60. This is the structure of Rho60 from the frog Xenopus laevis. Rho60 is a monomeric ring with a central cavity formed by alpha helical repeats. Our collaborators in the Reinisch lab also crystallized Rho60 with a fragment of Y-RNA that contains the high-affinity Rho60 binding site. This is a molecular surface representation of Rho60 colored by electrostatic potential. Positively charged surfaces are blue and negatively charged surfaces are red. The co-crystal with the part of the Y-RNA stem shown in yellow revealed that the stem binds on the outer edge of the ring. Binding is highly sequence-specific, with invariant amino acids in Rho60 contacting invariant nucleotides in the Y-RNA stem. Our biochemical analyses supported a model in which the remainder of the Y-RNA contacts this basic platform adjacent to the stem, allowing it to act as a gate to regulate binding of other RNAs inside the Rho60 cavity. So what RNAs bind in the Rho60 hole? Over the years, we had discovered that Rho60 bound misfolded RNAs in some animal cell nuclei. In Xenopus laevis ovaries, we found that Rho60 bound variant pre-5S ribosomal RNAs. And these RNAs were misfolded, inefficiently processed, and eventually degraded. In mouse embryonic stem cell nuclei, we found that Rho60 was complex to variant U2 acid RNAs that appear to be misfolded. And using cross-linking and high-throughput sequencing, Rho60 has also been reported to bind repetitive non-coding RNAs. So our biochemical studies revealed that Rho60 bound misfolded non-coding RNAs that contain both a protein-free single-stranded end of at least 5 nucleotides and adjacent protein-free helices. However, the specific sequences of the helices and tail were relatively unimportant. Our collaborators in the Reinisch lab crystallized Rho60 with a misfolded RNA fragment consisting of a tail and a helix. The 3' single-stranded end of the misfolded RNA binds in the central cavity and comes out the other end, while the adjacent helix binds to a large basic platform on the surface. Almost all interactions with the helix are to the RNA backbone, which explains the lack of sequence specificity. Based on these analyses, we propose that Rho60 scavenges RNAs that fail to assemble with their correct RNA binding proteins because they are misfolded. To learn more about the roles of Y-RNAs in Rho60, we have used bacteria as a genetically tractable model system. By studying Rho60 in the very first sequenced bacterium with an ortholog, Deinococcus radiodurans, we discovered that Rho60 functions with 3' to 5' exoribonucleases to assist their progression through structured RNAs. Most spectacularly, we found that Rho60 was tethered by Y-RNA to a ring-shaped exoribonuclease called polynucleotide phosphorylase, forming a new double-ringed RNA degradation machine specialized for structured RNA degradation. Using negative stain electron microscopy, our collaborators David Taylor and Hongwei Wang obtained this three-dimensional reconstruction of the RNP at 25 angstroms resolution. The reconstruction supported a model in which single-stranded RNA threads through the Rho60 ring into the nuclease cavity for degradation. Our biochemical studies supported a model in which Rho60 assists ATP-independent unwinding of structured RNA. These studies revealed another role for YRNAs, that of tethering Rho60 to effector proteins such as nucleases. So our bacterial studies revealed that Rho60 was bound to the Rho60 binding site on Y-RNA, while the exoribonuclease bound stem loops at the other end of the RNA, which we now call the effector binding domain. So what about mammalian Y-RNAs? What do they do? To learn more about Y-RNAs, Yuan Long in our laboratory used CRISPR to generate mouse embryonic stem cells lacking the two mouse Y-RNAs, Y1 and Y3. This is a northern blot demonstrating that she indeed generated cells lacking Y1, Y3, or both YRNAs. As a control, she examined cell lines lacking Rho60, and as expected, she found that YRNAs were reduced in the absence of Rho60. Yuan also found that just as YRNAs are reduced in cells lacking Rho60, Rho60 levels are reduced in cells lacking all YRNAs. She showed that she could complement the defect by expressing one or both Y-RNAs in the knockout cells. This is a graph. Rho60 is down by about 50% in the knockout cells, but increases when either Y1 or Y3 is expressed on plasmids. Rho60 is also restored to wild-type levels when a short RNA containing only the Rho60 binding site is expressed in the knockout cells. Yuan also showed that Y-RNAs are important for the cytoplasmic accumulation of Rho60. Here she performed immunofluorescence from wild-type cells and cells lacking both Y-RNAs. She found that in the absence of both Y-RNAs, there was reduced Rho60 in the cytoplasm and increased staining in nuclear foci, which she showed were nucleoli based on co-localization with fibrillarine, a nucleolar marker. And she was able to complement the change localization by putting back the two Y-RNA genes on the plasmid. She also showed that, similar to bacteria, Y-RNAs tether Rho60 to effector proteins in mammalian cells. She found that several proteins reproducibly co-purify with Rho60 from these cells. These proteins include MOV10, an RNA helicase implicated in structured RNA degradation, and ZBP1, an RNA binding protein that adapts the Rho60Y3 complex for nuclear export. By performing immunoprecipitations from wild-type cells and cells lacking one or both YRNAs, she showed that Y1 is required for the association of MOV10 with Rho60, while Y3 tethers Rho60 to ZPP1. Thus, in both mammalian cells and bacteria, one role of Y-RNAs is to tether Rho60 to effector proteins to create specialized RNPs. There are also reports that Y-RNAs are required for DNA replication in mammalian cells. This role is independent of Rho60 and involves sequences in the upper part of the Y-RNA stem. However, the fact that our mouse embryonic stem cells lacking Y-RNAs divided normally is inconsistent with the idea that these RNAs are essential for DNA replication. To examine this further, we compared the cell cycles of wild type, Rho60 knockout, and two Y-RNA knockout cell lines, since defects in DNA replication should slow the cell cycle. In these experiments, we measured incorporation of bromodeoxyuridine, a thymine analog, and used flow cytometry to separate cells by stage of the cell cycle. All four cell lines behave normally, indicating that at least in these cells, Y-RNAs are not important for DNA replication. In summary, I've told you that Y-RNAs folded to conserve secondary structures and bind the Rho60 protein. And binding of Y-RNAs is required for Rho60 to accumulate to wild-type levels and to localize to the cytoplasm. Y-RNAs also tether Rho60 to effector proteins to create specialized RNAs. Thank you.